Give me a second, and we'll start the webinar.
30 seconds, perfect.
You got the music, Dr. Ivan? You do?
Yep.
Perfect, I see it right here. And we'll start the webinar. All right, we're starting the webinar in 5, 4, 3, 2, 1. Our webinar has started.
All right, we'll just give it a minute for everybody to join.
Copy that.
Okay, great. Welcome everybody to this month's Miami Global Brain Tumor Symposium. We got really excited because we have a very special guest tonight. But before we start, just want to thank all of my co-directors, Dr. Komatar, Dr. Morko, Dr. Benjamin, and also Dr. Shah, who are my partners in crime here for brain tumor surgery and really helped put this together and help with insights on these complex cases. Also to our admin here, who help make this possible week in and week out now for over 2 years. Christina, Roberto, Ingrid, Damari, and Ignacio, really, we couldn't do it without you. Again, just as an update, all of this information can be found on YouTube on our University of Miami Neurosurgery site. We have over 10,000 views already on all of the videos that we've put up just for brain tumors. And then we also have the cerebral skull-base and vascular, pediatrics, and resident symposiums that are ongoing. And so you can find a ton of information there. Please be sure to check it out. Also, follow and share us on social media. So this week, we welcome again Dr. Shah, who's leading our immunotherapy here at University of Miami for brain tumors. And then our fellows, Dr. Barry, Dr. Morel, and Dr. Dagabati, who are joining us here to share their insights and questions from the audience. We encourage everybody to kind of use the Q&A button throughout the talk tonight, and we'll try to get to those during and after the talk. This month, we have an extra special guest, Dr. Higgins, who's joining us from his newly appointed position at University of North Carolina, Chapel Hill, where he's a major component of their brain tumor program and also is leading his own laboratory efforts on glioblastoma. Dr. Higgins has an amazing pedigree, did his MD, PhD at Columbia, did his fellowship here at University of Miami, has a tremendous amount of publications in both basic science and clinical work. He's working on several grants right now with the NIH, and I have no doubt he's going to be successful. He's an extremely talented surgeon, and I can't even comment on what an extraordinary year it was to have him as a fellow and just to see him kind of take the next step now to his own independence, and looking forward to seeing his success as a superstar in neurosurgery and specifically in brain tumors. So Dom, thanks for taking out the time and sharing with us a little bit about your expertise in this area and kind of what the future may hold.
All right, thanks Mike for the kind introduction. I'll start sharing. It's an honor to have a chance to share my work with the audience today. The symposium has been a great resource for us over the years, and I was glad to see it continue. I know it's a tremendous amount of work for you and the team, so thank you for continuing with it. So my talk today is shown here, targeting tumor metabolism, ferroptosis, and glioblastoma. I have one disclosures for patent on dietary-based ferroptosis treatment, but no direct financial disclosures. And so just an overview of what I'll show today. A lot of this work is started at Columbia, where I did my postdoc work with Peter Cano and Jeff Bruce and Brent Stockwell and in the group there. And we'll be carrying on here at UNC Chapel Hill. So we'll talk about what gliomas are and the role metabolism plays in tumor progression and malignancy. And then I'll introduce the concept of ferroptosis and the core components of the amino acids that really drive the process. And we'll touch on synergism with conventional treatments and then some translational strategies that we're gearing up to implement here and at other sites. And so glioblastoma, as most of the audience probably knows, the most common primary CNS malignancy. It's a type of glial tumor, which gliomas are a grouping of. And on the right, you see an MRI of what we likely describe as an aggressive splenial GBM that can be treated with different approaches. In Miami, we'd often do a laser ablation for these for good performance score preservation, cytoreduction, and to go on to treatment and clinical trials. But this is something that's not curable. Prognosis is, on median 18 months, but over the years we've seen evolving classifications based on molecular and histologic characteristics that drive that number up for some people, depending on what the actual tumor components are. And over the past decade or so, one of the biggest findings has been in the realm of metabolism. The enzyme isocitrate dehydrogenase has been a key player in stratifying tumors into by prognosis. And so the GBM microenvironment is heterogeneous with hypoxic components. The tumor grows rapidly, but eventually outstrips its blood supply. And so with this hypoxia, it results in shifts in what the cell metabolism has to do to compensate. And this is kind of speaks to the so-called Warburg effect, whereas the cancer cells have an increase in glycolysis, which is kind of the basis of PET imaging, which you can detect those changes. And then what you see is an increase in lactate production and a decrease away from oxidative phosphorylation, and pyruvate's not shuttling to the mitochondria anymore. And this is a study from a group at MD Anderson. They did a mouse model to look at radiation treatment of a mouse model glioma, and then they used a new technique called hyperpolarized MR spectroscopy, which is more sensitive than conventional MRI and can detect smaller changes in tumor metabolites. On the left, you see at the tumor at roughly about its peak, you can see an increased level of lactate, and pyruvate persists. After treatment, when the tumor regresses, you see that lactate level drop, and then during recurrence, that level starts to come back up, indicating that if we, we're able to track these signatures of the metabolites with increased accuracy, we'd be able to better track recurrence and response to treatment. Another study looked at different subclassifications of gliomas from GBM to more low-grade tumors and IDH status, wild type or mutated. And expectedly, there's differences in prognosis in the survival score at the bottom. But what we also see at the top is that the metabolite profile also tracks with the subtypes as well, with GBM having higher levels of carnitines, which are involved in fatty acid production and consumption. And so with that, it could start to see that something that targets metabolism is highly poised to be effective at the most high level from both a diagnostic and therapeutic standpoint. And ferroptosis was introduced in the past decade by a collaborator who discovered it, Brent Stockwell, was shown to be very effective in resistant cancers in their seminal paper with Dixon et al. You can see in this fibrosarcoma line, the cells are very responsive to a ferroptosis-inducing drug. And at the core, you see these components of alpha-ketoglutarate and pyruvate, glucose, and lipids that all interplay in this pathway. So what is ferroptosis? The definition is simply iron-dependent lipid peroxidation. So it falls under this umbrella of regulated cell death, apoptosis probably being the most widely known. It's independent of that and other regulated pathways like necroptosis is its own independent entity. And at the core of it is Fenton chemistry, which has a hydroperoxide reacting with iron to form these radicals. And these radicals can go on in the cell to interact with the lipid membrane, and then those generate lipid radicals. And then you have this second stage where that gets propagated and ultimately leads to disruption of the lipid membrane and cell death. The way to stop it would be to quench the radical formation with the termination, and that is due to antioxidants. In ferroptosis, that is accomplished by this enzyme at the core, glutathione peroxidase 4, which uses glutathione, an antioxidant, as a substrate to quench these lipid reactive oxygen species. So if you were to block GPX4, then you have unregulated lipid radical formation and cell death. And that's how you induce ferroptosis, by blocking GPX4. As you can see, this is a schematic from one of the reviews, it's a good read, of how all the components that go into the pathway. And then I'll highlight two of the key ones, which are the sulfur-containing amino acids. You can see down here cysteine, which feeds directly into the formation of glutathione, a tripeptide. And then upstream of that is methionine, which can be converted into cysteine by the transsulfuration pathway. So therefore, either targeting cysteine or methionine or GPX4 can result in inducing ferroptosis and can be potent from a therapeutic standpoint. And what others have actually shown is that glutathione levels correlate with GBM progression. So it speaks to the importance of this pathway for tumor progression. So there's different ways to target ferroptosis, and there's different classes of agents, and focus on class 1 and 2. Class 1 is a system XC inhibitor. So the cysteine can come into the cell by an antiporter, system XC or SLC7A11. And dietary cysteine is imported, and then that is converted into cysteine intracellular in exchange for a glutamate that's pumped out by this antiporter. That cysteine can then go on to be incorporated into GSH or glutathione, or dietary methionine, so essential amino acid, can be converted into cysteine, as I mentioned, by this transsulfuration pathway. These all converge on glutathione being formed and being a substrate for GPX4. And then you have a second class of drug, the GPX4 inhibitor, and this is like RSL3 or ML210. Those can inhibit GPX4 directly. It's a selenoprotein, and result in blocking any downstream effects from quenching the lipid radicals. And so sulfur amino acid restriction has been attempted by other groups. Specifically, methionine has been a very popular a target, and it's been shown to have promising effects in preclinical studies and shown to be effective for cancer therapies. There's actually a clinical trial that was unable to reach its target, but was attempting to show that methionine restriction in combination with chemotherapies can be effective in glioblastoma. More recently, cysteinase was trialed in pancreatic cancer and shown that depleting cysteine by this enzymatic method can be highly effective for these really aggressive pancreatic tumors. However, in the brain with the blood-brain barrier, it's hard to know if that would have the similar efficacy. So we decided to take a different strategy of using dietary strategies to try to deplete these precursors of cysteine and methionine and try to increase sensitivity to ferroptosis-inducing agents. And so we started with our cell models, and we had a number of different lines here, the 3 murine ones and 2 human ones, and then a non-glioma line. And then we saw different doses. RSL3 was exquisitely able to kill these cells at even lower doses. And then when we combine that with the cysteine methionine deprivation, we see that the effective doses dramatically decrease, and the cells have dramatically increased their sensitivity to the drug. And this is a quantification of that on the right. So then we tested whether or not RSL3 induces lipid peroxidation. And so to do that, we did a 2-photon assay with live cells. And then we treated them with RSL3 while undergoing imaging. And on the left, you can see that the uptake in green fluorescence is indicating lipid peroxidation is happening by a fluorescent agent called MODIP-PC11. And on the right, we have an inhibitor of ferroptosis, ferrostatin. So in the presence of ferrostatin, there is no lipid peroxidation indicating a specific reaction for ferroptosis. And this is just a timestamp of those images showing with high dose RSL3, you get a really fast onset of lipid peroxidation, which led to cell death after about 30 minutes. And that's completely rescued in the presence of the inhibitor. We then used flow cytometry, and we used a low dose of RSL3, and we saw a small response. But then when we added in the cysteine methionine deprivation, or CMD, that low dose of RSL3 was able to give the response of a higher dose, indicating that we had a more potent response or synergy. And then this is a quantification of that. In addition to using ferrostatin, you can see that CMD plus a low dose RSL3 is roughly equivalent to a high dose RSL3, which can allow you to get around some of the drug toxicity issues. And we tested to see if the GPX4 targeted effects are ferroptosis specific. We used another agent, ML210, to see if it would have similar efficacy as RSL3. And then we saw that on the left here in the blue curve. CMD plus ML210 was the most dramatic response. And then we added different rescue agents to see if other cell death pathways were playing a key role in RSL3 mediated cell death. And ferrostatin in blue was able to completely rescue the cells, whereas ZVAD, which inhibited apoptosis, and NEC1 that inhibits necroptosis, were unable to do that, indicating that it's specific to ferroptosis. We then used qPCR to check the hallmark transcriptional changes to see if they're occurring in our cells. And then we see CHAC1 and PTS2 and SLC7A11 are all upregulated with CMD alone. And so we did a proteomic metabolomic analysis of the cells, and see the light gray circle here on the left is a control, and then the right as the dark gray is, or the darker gray is the CMD. And we did principal component analysis, and you see there's clear separation of the populations in the two groups when we're looking at the metabolites that are affected. And not surprisingly, the highest level when we did a pathway analysis or cysteine methionine. And then also very effective is the taurine hypotaurine, which I'll get into in a little bit. As a heat map of that study, and you can just visually, it's a clear separation. And then we have our different agents that we can look into further in the future experiments. And so if you look at what happens downstream after cysteine gets formed, you have the taurine synthesis and salvage pathway, where cysteine gets broken down into hypotaurine and taurine. And so you would expect that any changes in the CMD diet would result in decreased cysteine as well as decreased taurine and hypotaurine. And that's exactly what we saw when we looked at the metabolites. We saw in the CMD group is decreased methionine, decreased cysteine, decreased hypotaurine, and decreased glutathione. And we did a fluorescent analysis to see what happened to reduce glutathione levels with treatment. And you see in the presence of CMD, we see a decrease in glutathione as well, which we'd expect. And then we did a seahorse analysis, which looks at extracellular flux to examine what changes are happening in oxidative phosphorylation or the mitochondrial respiratory chain. And we see essentially a global dampening of the energetic state of the cell with CMD, which may speak to the switch that's happening in the cellular metabolism. So we then injected mice with the tumor cells and then switched them to either control diet or our CMD diet. And what we saw is a modest but significant increase in survival with the diet alone without any additional drugs added on, which is encouraging. And so we then took those sections from the end stage mice, and then we used a spatial mass spec technique called DESI-IMS, or imaging mass spectroscopy. And then we could look at the specific region of the tumor and compare it to control matching sections and then the surrounding brain and see what are the specific metabolites that are affected. And what we saw is that the polyunsaturated fats and the different fatty acids that we'd expect to go into ferroptosis were the ones that were specifically affected. In summary for this first part, so that RSL3 induces ferroptosis in gliomas, and CMD, the cysteine methionine deprivation, enhances that ferroptosis and prolonged survival. And then CMD also induces specific lipidomic and metabolomic changes, which sensitizes the gliomas to ferroptosis. So then we had a collaboration with Simon Chang and his group, and led by his then MD PhD student, Ling Yi. And they were looking to see whether or not radiation itself can induce ferroptosis and be a non-canonical inducer of ferroptosis. And so we did that, and we looked at ex vivo slice cultures from GBM patients, and we treated them with one of those class 1 agents, IKE, which targets system XC. We combined that with a single dose of radiation, plus or minus the inhibitor ferrostatin. And we saw that the drug plus radiation had a significant increase in reactive oxygen species level by this DCF analysis, which is not specific to lipid peroxidation, but for reactive oxygen species generation. And that was inhibited by the presence of ferrostatin. And what was interesting was a shielding experiment where the nucleus can be analyzed separately from the cytoplasm. And then in the nucleus, you saw that the markers of traditional DNA damage that you would see from radiation were upregulated in the nucleus, but not in the cytoplasm. And that was specifically 4H&E, which is we've shown to be a marker specific for ferroptosis. So then we wanted to know whether or not CMD would synergize with radiation to induce ferroptosis, as radiation is a part of the standard of care for GBM patients. And so we used our same mouse lines, and we gave them either the diet plus or minus radiation. And we looked at a coefficient of interaction, the CDI score, to see if there was synergy. And across the two mouse lines and the human lines, we saw that it was below 1, it's actually closer to 0.5, which indicates a strong synergistic interaction. And similarly, we did a fluorescence analysis with flow cytometry to look at radiation plus the diet, and we see an increase in BODIPC11, a marker of lipid peroxidation, in the combination of radiation plus the diet. And then in a pilot study, we did a similar protocol to what we did prior with the mice, where this time we added on a radiation treatment in addition to the diet, and we found those to be the longest survivors of all of the groups. So now we want to know what our translational strategies are for implementing this as a ferroptosis-based therapy. And so what we're gearing up to do here at UNC in the coming year is to have this pilot study, a phase 1 study looking at safety, and we'll also test some efficacy in the tissue as well, whether or not patients on the diet show a metabolomic response during a tumor sampling and at the time of surgery. And we'll also do MR spectroscopy to see if we can non-invasively detect these changes as well. And then in the second cohort, given that we have shown that radiation can synergize with the diet, patients that are undergoing post-operative or radiation treatment for recurrent therapy, we'll combine them with the diet to see if there's any issues with safety, which we don't expect, and as well as efficacy. There's been studies in not in cancer patients, but in obese patients to look at more of a dietary strategy, and those were found to be very well tolerated. And in our mouse studies, we found no significant complications even up to 100 days. So we found that we feel like acute treatment would be well tolerated. And as you can imagine, something like this takes a large team that goes into it. We'll be working closely with the Lineberger Cancer Center, which has been amazing. Also, UNC has one of the largest nutrition research institutes with a powerful team that is capable of doing some really in-depth metabolomic analysis with a great clinical nursing staff as well, in addition to the Department of Neurosurgery and Pharmacology. And then so beyond that, what we want to try to do then is implement our treatment strategies in patients. And so in the preclinical setting, what seemed to be the most robust drug of choice is RSL3, which is this GPX4 inhibitor. So ideal long-term would be to combine the diet with delivery of RSL3. Given the issues with blood-brain barrier penetrance, and we have to figure out, how can we get this drug into the brain and into the tumor. And so what we did, we tested a similar paradigm in the mice where mice were injected with the tumors and then placed on the diet. And then we placed convection-enhanced delivery pumps in the mice and then assessed for survival after an acute treatment with RSL3. And again, we saw that most significant survival was in the combination treatment of the drug plus the diet beyond all the other conditions, which is promising and encouraging to going forward with clinical trials. And so for future directions, what we want to show is that there's clinical safety and efficacy of CMD in glioma. And then we touched on some of the combinatorial therapeutic strategies that we're looking into. We're also working with the labs here at UNC. They have a really robust center for small animal surgery and expertise in stem cell-based delivery of pharmaceuticals. So we're planning to do stem cell-based honing strategies to combine with ferroptosis agents to see if we can also use that strategy to target the tumor cells specifically. And then we focused on GBM for this talk, but there's a, the question is whether or not this would hold up in recurrent gliomas, which has been shown to have a switch in their metabolomic profile in terms of subclassifications. They tend to resemble more mesenchymal tumors and tend to be more resistant, and they've undergone treatment already. So we're doing our pilot studies now in those settings to see if we have similar efficacy. And also what's the metabolite metabolic landscape that changes with a recurrence. And then likewise, whether or not this would be efficacious in low-grade gliomas, the presence of IDH mutations could argue could either be for or against. Our preliminary data would suggest that they are, as sensitive or more sensitive than the GBMs as well, which speaks to the power of the strategy in terms of targeting tumors that don't need to be simply highly proliferative, but just targeting the metabolic profile. And so thank those that involved, a lot of people that helped out with this project. I want to specifically point out Pavan is now a resident at Columbia in his second year, who was invaluable in terms of completing this project. And Angelina, who's a co-author on this paper as well, who completed a lot of the cell modeling and the convection enhanced delivery studies, and Thanasis, who helped out with the live imaging as well. And then Matei Banu, who's a rising neurosurgical oncologist, who also has a strong interest in ferroptosis and immunotherapy as well. And then the Stockwell Lab for their ferroptosis, and Simon Chang and his group for radiation, and Marcus Sigeling for the Seahorse, and Peterson for the analysis, and also the UNC team here. And so I take a moment to thank my old family in Miami. It was talking with Mike before we went live, and just saying what a great year it was. We worked hard and doing 4 to 6 cases a day, but you don't feel it just because you're having so much fun, and a great group to learn from and to spend a year with. I highly recommend it to anyone that's applying for neuron fellowships now. Couldn't think of a better place to hone your skills in a great environment. And there's also the international observership that you can do as part of the program. And thank Professor Froley for letting me spend part of the summer in Paris with him. I learned a lot from their group and getting to see how they tackle similar processes over there. So that was awesome. And Mario Kart as well. So and this is where I do my grant writing these days. A nice view of the tower, and is my Instagram handle if anyone's interested in a research position or coming down to check out the program. Feel free to send me a message. And with that, I'll take questions.
Awesome. Thanks so much, Tom. That was great. Can you comment a little bit about, GBM is so heterogeneous. What are the kind of the spectrum of these metabolic changes that happen both intratumorally and between tumors? I mean, we know their mutation status, but do you think that there's even more changes between IDH within the subgroups, within the IDH1 mutants and whatnot, and then within the tumor itself?
Yeah, it's a great question. I think the IDH mutation was identified about around 2009, and I think initially the perception was this was something we happened upon that may be a bystander effect of all the other mutations, versus whether or not it was something that was driving. And I think what I think I'm seeing and other groups are seeing is that it actually plays a role in terms of like what's happening within the tumor. You have like production of oncometabolites, and that's going to lead to intrinsic changes within the cell. So in that paper I showed there's, you know, a separation between tumors with IDH mutations and IDH that don't, IDH wild type tumors, in terms of what's primarily affected. And so I think when we talk about metabolomics, it's kind of a broad umbrella of things, but you have, you know, changes in lipids and you have changes in more like the carbohydrate metabolism. And I think that's part of the separation that you'll see as well. Some tumors that may be more IDH mutated or may affect more the TCA cycle, just given the location of it, whereas IDH wild type tumors may have more of an effect in terms of lipid metabolism. But all that I think is what we're still figuring out. And I think you know, there's different ways to come at it. You know, serine metabolism is shown to be very, very promising in terms of a target. And there's a lot of interest in like ketogenic diets, which, you know, Alexis and I have talked about ad nauseum over the year. And so there's a lot that goes into it that we're just kind of on the cusp of figuring out. But I think it'll be very informative in the coming years.
Alexis, what questions do you have?
Great talk, Dom. Really, really interesting. I'm looking forward to hearing more of these interesting projects that you have over there. You know that this is an expanding, you know, like field. There are like so many combinations that, you know, you can think about, you know. So great projects. So congratulations. I have two straightforward like questions. The first one is actually if you have seen any like upregulation of like alternative pathways, like metabolic pathways that GVMs can use to increase the tolerance to CMD.
That's a great question. So what what others have found is that you know, a chronic diet may not necessarily be the best strategy because there's systemic compensation that happens primarily like from a you know, kind of liver and systemic standpoint. Eventually, if you deprive the body of cysteine methionine, it's going to kind of produce its own, right? So you have a catabolism that's happening that's going to kind of replenish the supply. So when we do the studies, when we look at essentially like a time course of the acute versus the chronic, and we see that there's a dip that happens that's around like, you know, in that early week period, but then the levels start to come back up, which kind of speaks to the body compensating for what you're doing. So in our animal studies, we did a chronic diet, which we found was effective. But when we do an acute treatment as well, we do see a significant metabolic response, and potentially we'll see a good survival response as well, which would be great for translational strategies, because one of the biggest issues with trying to implement these dietary-based strategies is compliance. So if the shorter you can make that window of what your treatment needs to be, the more patients will be able to stick to it. So that's what we're seeing. And so one of the things I'm delving more into is how can we optimize this acute strategy, essentially get the most bang for your buck without kind of dragging on how long you need to be on a specific therapy.
Perfect. Awesome. So I think that it's going to be like more like a stressing the tumor like acute.
Exactly. Yeah. And historically, you know, I think I lost you for a second. So yeah, so historically, you know, starving the tumors, you know, already been shown to be, you know, effective. If you don't eat, your tumor will die, but you know, so will you. So you have to find some middle ground where you're primarily affecting the tumor without hurting the patient. And so one of the things we also did was we tested normal astrocytes and we tested the surrounding brain, and we show that the response, and especially in the acute treatment, is limited for the most part to just the tumor without affecting the surrounding normal cells. So which is encouraging for moving forward.
Perfect. I have another question. So do you think that this can have like a role in other type of tumors outside the CNS?
Yeah, definitely. You know, the cysteinase experiments in the pancreatic cancer are promising. So I think you know, ferroptosis has been, if you look at kind of the publication like tracking over the past, you know, 5 years, it's kind of gone up exponentially. So it's been looked at in a number of the cancers. I mentioned fibrosarcomas, pancreatic cancer, and renal cell is another potential avenue. So I think it's not just limited to CNS cancers, and it's actually not just limited to highly proliferative cancers. I think it's about what's the intrinsic state of the tumor, and then what makes it even more powerful is that if the tumor is not very sensitive, and you can have a strategy to sensitize it, then you've kind of broadened the what you can treat with. So
Perfect. Thank you, Tom. Great work.
So if you look forward to like at a potential clinical trial for this, I mean, how do you keep track of that? These tumors could respond. Is this a blood test? Is there a biomarker, or is there something you could sample along treatment that you could say that these are causing stress and you're changing their?
I think from the diet itself, just to see if when we're in that responsive phase, you can do serum markers, and that's why I kind of point. That's why taurine and hypotaurine are pretty valuable, because you can do serum tests for those, and you can also test things like glutathione levels, cysteine and methionine. Some of those things that the pool of active metabolites doesn't shift that much, so then it becomes a little bit less sensitive. But I think taurine and glutathione are some of our more promising targets, and that's where mass spec comes in. Imaging mass spec comes into play, because then you can non-invasively screen, because you're essentially doing an MRI to detect metabolites, and there's already protocols to check metabolites of cysteine to test glutathione in patients, and then the hyperpolarized MR spec, which uses like carbon-13 labeling. You can specifically track different components of the Krebs cycle, and then with PET imaging you can do labeled methionine. So that's another aspect that I'm getting into with my lab. Is we're partnering with NC State in a group here, and we have a new hyperpolarized MR spec machine that we're going to be testing on our samples to see can we detect some of these changes non-invasively. So that gives us a perfect readout for what's happening real time as we're doing therapy.
And my last question is, and this may just have to do with the fact that, throughout the world, we can't even decide on what is the right diet for us to live normally without cancer, which is crazy when you think about how much science we know, right? But there's so much debate on what is the best diet to both live, to maintain weight, to lose weight, to maintain muscle. But why do you think in cancer there still is no real recommendations on what is the best diet, and how do you sensitize these tumors to these responses? Why do you think it's taken so long to kind of figure this out? Any thoughts?
Yeah, I think the short answer is that I realize it's complicated. The more you look at it, the more you see that these are all there's an interplay going on with all these amino acids, these metabolites. So I'm not trying to say this is the sole solution from a dietary standpoint. That's the only pony that has a potential to win the treatment race. But I think you have to think about it in terms of how do you want to implement it. I think anything that's exclusively a dietary strategy has a lot of shortcomings, just from one, the compliance standpoint, and two, a compensation strategy. So you have to. I look at it as what's your combination treatment. What's your diet helping you to do otherwise? Because the effect of the CMD in that experiment I presented, there's room to optimize it and enhance the effect, but it's a modest effect. But when you combine it with either radiation or a GPX4 inhibitor, then you get a more robust survival profile. So I think it's about it's like doing a multi-drug study. Like you want to say what's our synergistic strategy. If you're going with something that's a ketogenic, why are you doing that? Is that because you think it'll help with radiation, or if you have a new drug that you think it'll target, it'll help with imaging? But I think that's where this is so promising, is that essentially CMD gives you what a class 1 ferroptosis inducer does, which essentially blocks the antiporter. It decreases the amount of cysteine available, and then you're coming in with a really potent drug. This is a GPX4 inhibitor, and that's part of the reason I got so interested in this project when I started in the lab, and I saw the first study where we added the drug and cells were just gone. You add a rescue agent and they're there, which is kind of the perfect phenotype for something that's an effective therapeutic strategy. And you think of like the availability of lipids in the brain, it just seemed like it's a winning combination. So we're excited about kind of digging into this further and trying to see if we can get this translated. But I think that it is complicated, but I think our combination strategy is what will give us the best benefit.
Right. And we have one last question here from our medical student, Freddie Vallejo, who's also been working on some metabolic activity in the lab. He wants to know, is there a reliable protein to assess via Western blot for ferroptosis that's analogous to, that's similar to the cleave caspase assessment for apoptosis?
You could, it may just be a bias standpoint. We don't do a lot of Western blots in our experiments. As you can see, 4-H&E is pretty reliable, but it's we've kind of gone at it more from a immunofluorescence standpoint. But there's some markers that you can use, cellular markers that like 4-H&E and some others that will be able to give you like a readout. But our go-to has been using a lipid peroxidation marker, because that's the definition of it. So you have something like PODP-C11. If you see it turn on, it's lipid peroxidation. There's no question about it. You can't always do that because of some experimental limitations, but that's been my preference to use. DCF is a good substitute if you have some issues with PODP, but it's less specific, but it can be reliable as well. And then qPCR is also very reliable. If you have your signature transcriptional changes, CHAC1 is a pretty reliable marker of acute ferroptosis is what I'm seeing. But also SLC7A11 can be a good marker as well, but you just kind of have to screen your own cell population before you go forward. And then lastly, the glutathione assay is kind of a hallmark, just because it's a little different than apoptosis. And so I think looking at that from looking at it from that standpoint is a little more helpful.
Awesome. Well, again, congrats on the work thus far, and we wish you the best of luck as you take this to the next step. I have no doubt you'll be successful, and we expect answers within a year.
So do other people here.
Congrats again, Tom. Great talk.
Thanks.
Hope all is well.
Guys.
Stay safe, and we'll talk soon. Okay.
All right. Take care.